Okay, so as we learned earlier, when Watson and Crick figured out the structure of DNA and built their model of DNA, they realized how DNA could be replicated. And replication is a little bit different in prokaryotic cells versus eukaryotic cells. And that's something we haven't learned about yet, but prokaryotic cells are cells that don't have a nucleus, bacteria. So when you think prokaryote, think bacteria. When you think bacteria, think prokaryote. All bacteria are prokaryotes. The other kind of cell are cells that have a nucleus, like your cells, and those are called eukaryotic. So this is a prokaryotic cell. The DNA is found in the nucleoid region. So they don't have a nucleus, but they have a region where the DNA hangs out, where the DNA is found, and that's known as the nucleoid region, not nucleus. The prokaryotic chromosome is different from a eukaryotic chromosome in that it's just one DNA molecule, circular, so it's a ring of DNA, and it's uncondensed. Now, you're not going to be familiar with what that means, but it basically means it's not all coiled up and packed together into something that takes up less space. Basically, that's what the condensation does in a eukaryotic cell. But this is a prokaryotic cell, so this DNA is uncondensed, and finally, it's not associated with proteins. In a eukaryotic cell, the DNA has all kinds of proteins along with it that cause it to condense, that pack it and condense it. So histones are the names of those proteins. So in a prokaryotic cell, there are no histones. There are no proteins associated with the DNA. So this is the nucleus of a eukaryotic cell, not the whole eukaryotic cell. This is only the nucleus of a eukaryotic cell. That's what this diagram represents. And eukaryotes have about 1,000 times the DNA that prokaryotes have. Prokaryotic cells are much smaller, about 10 times, just on average, about 10 times smaller than a eukaryotic cell. But they have 1,000 times less DNA than a eukaryotic cell. Eukaryotes are more complex. Eukaryotic cells are more complex. And so that's the reason there's more DNA. The DNA can be found as uncondensed chromatin in non-dividing cells. So notice that in this diagram, this kind of shaded, the whole interior basically, that's not some other structure, is shaded. And that's representing chromatin. And we'll learn on the next slide what that's really referring to. But it's uncondensed DNA in the form of chromatin. And that's what you find in a non-dividing cell. So you don't see what's labeled here chromosomes in a non-dividing cell. In a dividing cell, that's when the chromatin condenses into chromosomes, and you see the actual chromosomes. And we would refer to these as mitotic chromosomes because mitosis is involved in cell division. So in a cell that's about to divide, the chromatin condenses into mitotic chromosomes, and those are then going to be divided up between the two new cells when the cell divides. And that's really the whole purpose for that condensation. The whole purpose is to condense the chromatin into chromosomes that can be easily segregated to the two new daughter cells, as we call them. So what's wrong with this diagram is that you don't see chromatin and chromosomes at the same time in the same cell. So if we contrast the eukaryotic DNA with prokaryotic DNA, prokaryotic is single, whereas in eukaryotes there's more than one chromosome. For example, humans have 46. In prokaryotes, the chromosome is circular. In eukaryotes, it's linear. So each one of these two, as we call them, sister chromatids making up one chromosome, each one of those chromatids is one molecule of DNA, and it's linear. It's not a ring or a circle of DNA, it's linear. So there are two molecules of DNA in this mitotic chromosome, but they're linear. Condensed as opposed to uncondensed. So in prokaryotes, the chromosome is not packed and coiled and condensed, as we say, and not even at the time of cell division does it do that, like it does in eukaryotes. And then the fourth thing is that eukaryotes have these proteins associated with the DNA that bind to the DNA called histones. And again, that'll become more clear on the next slide. But that's just a contrast between the form of DNA that you find in prokaryotic cells and the form of DNA, or the forms of DNA, that you'll find in eukaryotic cells. And then the last point here is that different species have a different number of chromosomes. So for example, we have 46, chimpanzees have 48, our close relative, but we don't have the same number of chromosomes. Different species have a different number of chromosomes. And there's no relationship between the number of chromosomes and the complexity of the organism. For example, trees, or plants, I should say, can have hundreds of chromosomes, and we'll be discussing why when we move along here. So here's where we see what's going on with the DNA and its association with these proteins called histones, and how the DNA wraps around the histones and then coils into chromatin, and which then coils into chromosomes, finally into these mitotic chromosomes. So histones are proteins that organize the DNA into chromatin and chromosomes. So that's basically the definition of a histone. A nucleosome is the DNA when it wraps around a cluster of 8 histone molecules. So if you count up the number of molecules in each one of these clusters, you'll find that there are 8 histones. And then the DNA wraps around that cluster of 8 histones. And that's known as a nucleosome. So that's a nucleosome, that's a nucleosome, nucleosomes. And chromatin is DNA organized as nucleosomes. So in other words, as soon as the nucleosome clusters, the histones cluster into, as soon as the histones cluster and the DNA wraps around it, that can be considered DNA. So the histones here, can be considered chromatin. So these nucleosomes here that aren't really packed together at all, or close together at all, that is considered chromatin. But so are these, the 11 nanometer fiber here, where the nucleosomes have kind of bound together, and then when they coil up into the 30 nanometer fibers, that's considered chromatin also. So everything from the 30 nanometer fibers down to the 11 nanometer nucleosomes is chromatin. One DNA molecule wraps around histones to form 11 nanometer nucleosomes, which coil into a 30 nanometer fiber. And again, that's chromatin. And then it doesn't do this until it's time for the cell to divide. That's when the chromatin, which again ends here at 30 nanometers, supercoils into these 300 nanometer fibers, which then pack and condense even further into 700 nanometer chromatids. So this is one sister chromatid, and this is another sister chromatid. So they don't cross each other, they're side by side, they're connected side by side, and each one is a DNA molecule, and each one has coiled and condensed into a mitotic chromosome, making up one full mitotic chromosome. And so that would be 1400 nanometers. So chromatin is the working form of DNA. In other words, this is when DNA can actually function it can replicate and it can be transcribed into RNA. DNA condenses into mitotic chromosomes only for cell division in order to be orderly segregated into 2 new daughter cells at the time of cell division. And so that process is the process of mitosis and cell division. And just to ease your mind a little bit, you don't need to know all of these different nanometer sizes of the chromatin and the chromosomes. Really, all you need to know is that the DNA wraps around histones to form nucleosomes, and that the nucleosomes then pack into chromatin, and the chromatin then coils and condenses into chromosomes. So because the chromosomes are different in prokaryotes and eukaryotes, DNA replication is a little bit different in prokaryotes versus eukaryotes also. So the prokaryotes have a circular chromosome, and that's what's being represented here. It's also being represented in this diagram. So this is a cell that's undergoing cell division. This is a prokaryotic cell undergoing cell division, which in prokaryotes, because they're single-celled organisms, is the same thing as asexual reproduction. So when a single-celled organism divides, it's reproducing. But for all cells, no matter prokaryotic or eukaryotic, the DNA has to be copied first. The DNA has to be replicated first before the cell actually divides and splits in two. So in prokaryotes, because it's a circular chromosome, that happens by starting in one place, and that's known as the origin of replication. I'm trying to catch up here to where the text is. So, yeah, like it says here, in prokaryotes, replication starts at the origin of replication, and there's only one in prokaryotes. That's different in eukaryotes. And the DNA unwinds and unzips at the origin of replication, and new DNA strands or new DNA polynucleotides are built on the old polynucleotides, and that proceeds in both directions around the chromosome until it meets at the other end of the chromosome or at the other side of the circular chromosome. And that is a point known as the termination of replication. So we've got the origin of replication here being labeled with an ORI, and we've got the termination of replication at the other end. And that's really all you need to know. You don't need to worry about all these other terms that are on here. And that because it's a circular chromosome, replication proceeds in both directions until the replication forks, as we call them, meet at the termination of replication. And this is referred to as a replication bubble. But all of this is regulated by enzymes. So how does this all happen? How does the DNA unwind and unzip? How do new nucleotides get added to the old strands of the DNA? It's all enzymes. Those protein molecules that I've told you before pretty much run the show in living things. So as we learned on the previous slide, eukaryotes have about 1,000 times the amount of DNA as prokaryotes, and there are multiple chromosomes, and the chromosomes are linear. The DNA molecules are linear as opposed to being a ring. So replication has to happen a little bit differently, and really the major reason for the difference, or the major difference, is that there are multiple origins of replication and multiple replication bubbles that have to form in any one DNA molecule. So there can be thousands of origins of replication on a DNA molecule, because DNA molecules can be millions of base pairs long, as we say, millions of nucleotides long. But the replication itself is the same. In other words, there's a replication bubble, and on either side of the replication bubble there are replication forks, and new nucleotides are added to the old polynucleotides to produce a new polynucleotide in both directions. That's taking place. And eventually these replication bubbles, as this diagram is trying to show, eventually the replication bubbles run into each other and merge together. And when that happens, replication is complete. And in this diagram they're showing that the old strands are dark blue and the new strands are light blue. So every DNA molecule following replication has an old strand and a new strand. And the new strand was built on the old strand, and that's why it carries the same information. It's complementary to the old strand. The new strand's complementary to the old strand because the nucleotides were added according to the base pairing rule. So there's no way for the information to change as long as there are no mistakes. Mistakes being mutations, which can happen. But there are mechanisms to prevent that from happening. So this is all basically explaining what I just explained. These two molecules here that result from the replication from one DNA molecule, though, represent sister chromatids. Again, in eukaryotes, this is one mitotic chromosome, and it's made up of two sister chromatids, and those two sister chromatids are identical to each other because they're the result of replication from a parent DNA molecule. And of course, this is all regulated by enzymes. And then finally here, there's this inset photo transmission electron micrograph of DNA replicating. And we see 3 replication bubbles here. And at either end, the arrows are basically pointing to the replication forks. But the reason these replication bubbles are different sizes, different than what we see in the diagram here, they're all the same size, and that they were just trying to simplify the diagram. But these are different sizes because they have been replicating for different amounts of time. This one's been replicating longer, whereas this one's in the middle, and this one has been basically just started replicating. But eventually they'll merge together, and so it doesn't really matter whether they started at the same time or not. So if we were to zoom in on a replication bubble, this is what it would look like. And hopefully you're relating this to what you're working with in the lab, with the paper models in the lab, because what you did during replication is the same thing that's shown here. So replication begins at the origin of replication, which would have been about right in the middle of this replication bubble. And 2 replication forks are formed by enzymes, an enzyme called helicase. And remember, any good name of an enzyme describes what it does, and it ends in ase. So helicase unwinds and unzips the double helix. Good name for the enzyme. But it's not shown in the diagram here. You could draw it in. It's operating right here where the replication, where the DNA is being split into a fork, into the replication fork. And over here too. So it's operating at both ends. It's operating at both replication forks. Again, it's called helicase. But then DNA polymerase, which is the big player in DNA replication, and so these blue spheres are representing that. You see that DNA polymerase is adding new nucleotides to the old nucleotides on the old strand. So the old strands here are being shown in dark blue, and the new strands are being shown in red. So again, during DNA replication, new strands are built on the old strands, according to the base pairing rule, A with T, G with C, right? A, T, G, C. And that ensures that the DNA is faithfully copied without mistakes, usually. But DNA polymerase actually has like a proofreading ability, so if there is a mistake made, it will actually fix itself. So it enforces the base pairing rule by proofreading. But just as we saw in the previous slide, this is going to continue in both directions, and there are other replication bubbles in this DNA molecule that are replicating, and so they're all going to merge together and the DNA will be copied, the whole length of the DNA molecule, which again can be millions of nucleotides long.